So tonight's topic is about chromosomal inheritance and how that can help with genetic variation and how if that goes wrong, we could get certain disorders and diseases, hopefully based off of what we've already talked about with meiosis and things you have seen before, including in your freshman year, that this will be mostly review. Okay, since our focus here is on chromosomal inheritance, we're going to talk about things right now that increase genetic variation, dealing with the entire chromosome. So when we did meiosis, we talked about crossing over, independent assortment, and how those things caused variation. We're not mentioning crossing over here, okay, because we're talking about the entire chromosome right now. But remember, it does still increase genetic variation. So these three things are going to help increase genetic variation via sexual reproduction. And the first one being segregation. Remember, when the gametes are made with meiosis, the alleles for each gene move away from one another, right? And they don't care about the others. So you can see this happening here, where the alleles for the genes are going to be separating from one another. So that would lead to variation. Independent assortment, where our non-homologous chromosomes assort independently. So happening here, we see these chromosomes lining up on the metaphase plate, okay? And we don't care. They're going to assort independently, okay? They are not, you can see how there's two options here with how they lined up there. They do not care what the other pair of chromosomes are doing. So they segregate independently, so the alleles are separated, and then how the non-homologous chromosomes line up on the metaphase plate is also independent. So what pair 1 is doing does not affect pair 2. And then fertilization also can affect genetic variation. So the gametes, to form the gametes, we've already done segregation, we've already separated the alleles, we've already done independent assortment. So we've already incorporated some variety within the gamete itself. And then the combinations of those gametes can give us even more variety there. So I get different allele interactions based on what sperm and what egg interacted. So chromosomal inheritance is also what can provide us and teach us about the patterns of inheritance from parent to offspring. So for example, humans and other mammals have two types of sex chromosomes. We have an X and a Y. Remember, so let's go with humans as our example, right? Remember, humans have 23 pairs of chromosomes. Pairs 1 through 22 are what are called autosomes, and then pair 23 is our sex chromosomes. So pair 1 through 22 contain genes that code for pretty much everything in our body, and pair 23, our sex chromosomes, will help us determine sex or gender. So in females, there is 2 X chromosomes, and in males, it is an X and a Y. So in mammals, in the testes and the ovaries, the 2 sex chromosomes, they'll segregate during meiosis. So what that means, when I form an egg cell, right, when a female forms an egg cell, every egg is going to end up with an X, right, because she doesn't have anything else to segregate out. But when males form sperm cells, some of the sperm cells will end up with an X, and some of the sperm cells will end up with a Y. And this becomes important if we have specific disorders that are linked to these particular chromosomes. So the pattern of inheritance here for these sex-linked chromosomes can cause certain genetic disorders. So for example, let's look at X-linked genes. So X chromosomes carry genes for lots of traits that are unrelated to sex or to gender. Okay, so not everything on the X chromosome is all about sex and gender. So if the trait is linked to the X chromosome, so if the trait is carried on the X chromosome and it's a recessive trait, females have to get both copies of that trait to get the disorder. Right? But males, because they only have one X chromosome, if they get one copy of the recessive allele, then they express the disorder. Their option is either to express it or to not. There's no carrier situation in the males. So if the male receives the allele from his mother, he will express that trait because the dad is who always has to donate the Y. This had to come from dad because even though dad carries an X, mom does not carry a Y. So the only way a son can get a Y chromosome is from the dad. So the mom could pass on her X-linked gene to the son, and the son could express the disorder because there's no carrier, there's no heterozygotes, there's no homozygousness in this. It is just one allele, so that's what you have. For example, color blindness. Color blindness can affect women, but it's not nearly as common in women because women have to get both recessive alleles. While we're on this, let's look at some Punnett squares involving sex-linked traits again. Remember, so I only need to use X and Y if I know the trait is sex-linked. And so that is what trips most people up. Again, we did these your freshman year, but this is what would trip most people up, is whether it was sex-linked or not. So if it's a known sex-linked trait, for instance, color blindness or hemophilia, then we would need to use our X and our Y. So let's say we had a mother who was a carrier. So she's heterozygous, big H, little h, but she is not colorblind. She is a carrier. And the dad is not colorblind. Well, that tells me the dad automatically is dominant because it's an X-linked trait. So I want to know the probabilities of their offspring. So I can draw my Punnett square. My gametes for my dad is that he can pass on the X or the Y. Gametes for the mom is that she has 2 possible X chromosomes to pass on. So I have one female option who would not even be a carrier. I have a male who would not have color blindness. I would have a female who would be a carrier, but she would not be colorblind. And then I would have another option for a male who would be colorblind. And so based off of this, maybe you made the leap or you started to make the leap. Well, how could a female ever be colorblind? Well, for a female to be colorblind, her dad has to be colorblind. Her dad has to have that recessive trait. That's the only way that she can get, right? If mom can be a carrier, mom doesn't have to be colorblind. But for a female to be X little h, then there has to be an X little h that each parent can give her. So her dad would have to be colorblind, and the mom, at minimum, would have to be a carrier. So it's not as likely for women to develop these X-linked disorders as it is for males. So if we applied our probability rules here and we wanted to know the probability of a male that is colorblind, well, the probability of a male is 1/2. The probability that they are colorblind, okay, looking at the males here, is 1/2. And so I get my 1/4. I want those two things to happen at the same time so I can multiply them, right? And that is true based on my Punnett square, right? 1/4. So let's look at another disorder here, sickle cell anemia. So sickle cell anemia is a substitution of a single amino acid in hemoglobin. So hemoglobin is a protein. Remember, proteins are lots of amino acids strung together. And so for sickle cell anemia, you can see, maybe I shouldn't do it in red so you can see it better. Okay, you can see there's normal and there is the mutation there. So I have a substitution mutation that has caused a single amino acid to be different or be changed in the hemoglobin. This is a form of recessive inheritance, which means both parents have to be able to pass this on to their child. Okay, so either both parents need to be carriers, right, so that they can pass on little s's. One parent could have it, but then the other one has to be a carrier. Both parents could have it, right? But this is a recessive inheritance. So it deforms the red blood cells into a sickle shape. And the sickling shape causes multiple effects, that pleiotropic situation, right, where I've got one change that's causing lots of effects, a cascade of symptoms. You can see over here on the right, these sickle-shaped cells, they cause weakness, anemia, heart failure, pain, fever, brain damage, spleen damage, damage to other organs. So one issue here is causing lots of different effects. So that's our pleiotropic situation. This is another example, too, of structure and function. right? Normal red blood cells are built in this particular shape that helps them carry oxygen as well as helps them fit through the capillaries. And so when this sickle shape happens, they clog up the capillaries. Okay, and then blood flow ceases to go to certain places. So another example of structure and function. So what about entire chromosomal changes where we end up with the completely wrong number of chromosomes? Okay, so this is a non-disjunction mutation. Okay, so non-disjunction, so the junction, they didn't separate properly. Okay, that's what this is going to mean. They did not separate properly during meiosis. So what will happen here is I'm supposed to have pairs of chromosomes, but I could end up with just one, or I could end up with a set of 3, and we'll talk more about this in just a minute. You also could have changes to the actual chromosome that involve a deletion. So I have my chromosome, right, and the change ends up being where a piece of it is completely missing. Okay, and so that would be a deletion, so my chromosome is going to get shorter. I could have a duplication issue where I've taken my chromosome and then I've inserted a whole other copy. So my chromosome gets a lot longer, and I have whatever was added is a duplication of what was already there. And these are not beneficial. When we are changing the lengths of the chromosomes, that is a problem. Okay, I could have an inversion. And hopefully these are familiar from your freshman year, right? I could have 1, 2, 3 genes, and I could take them out, and that should be a 1. I could take them out, flip them over, reinsert them back in the chromosome. Shouldn't have gotten bigger. It should have stayed the same size, but I just reinsert them back in the chromosome, but they're in the wrong order now, so that's not good. And then a translocation always has to involve 2 chromosomes. I'm going across to a whole different location. So I may have had chromosome 1 here, okay, but I'm actually exchanging information over here with chromosome 4. So now I end up with kind of like crossing over between 2 non-homologous chromosomes. Okay, and so that is a problem. I cannot switch pieces of non-homologous chromosomes. These are major, major mutations that could happen to the chromosome. So I'm missing part of it. I duplicate parts of it. I take part out of it, flip it over, put it back in. So I invert it, or I switch with a non-homologous chromosome. Non-disjunctions, again, are also a major mutation. So non-disjunctions are a result of a problem with the spindle during meiosis, and so the daughter cells are going to have errors. So there's 2 different ways this could happen. The homologous chromosomes don't separate properly during meiosis 1. So you can see this happening right here in this first one. So these homologous pairs right here did not separate properly. So you can see how in the daughter cells, they do not have the same number of chromosomes now. Or the sister chromatids could fail to separate during meiosis 2. So you see that happening over here in the second one. Okay, so I've got these sister chromatids here that are not separating. So I end up with a gamete that has 3 and one that has 1, instead of 2 gametes that each have 2 like I should. So what happens here is the baby will end up having the wrong chromosome number. So these gametes, they don't get trashed. So let's say this is an egg with 3 chromosomes or a sperm with only 1 chromosome. They could still participate in fertilization. And so the baby will end up with the wrong number of chromosomes. So the monosomy is right here, our N-1, okay, and is a result of our N-1, and our trisomy is a result of our N+1. So let's look at an example of these. So trisomy 21, or Down syndrome, okay, we can see how right here at pair 21, there is, instead of pairs of chromosomes, it is a set of 3. If it was a monosomy, instead of having pairs of chromosomes, somewhere there would be, like for example, like this, there would be only 1 chromosome. So a trisomy has 1 pair of chromosomes that's a triplet instead of a pair, and a monosomy will have 1 set of chromosomes that is 1 instead of a pair. So we're going to continue practicing with, we will start introducing again now into your problems. You'll learn to see sex-linked traits showing up in your Punnett Square practice problems now. You guys will be bringing in your genetic research posters so that you can look at some other examples of these diseases and stuff too as well.